Hello friends, hope you all are well and doing good. Welcome back to my channel. In the series of biotechnology and its principle, this is my 8th video and today I'm going to talk about insertional inactivation. But before we start our topic, let's have a quick recap on what we have started earlier. Now we all are familiar with the process of recombinant DNA technology. In the process of recombinant DNA technology, our gene of interest, or you can say a foreign DNA or alien DNA, is inserted into a vector DNA or cloning vector with the help of the enzyme like restriction endonucleases and ligases. This will result in the development of a recombinant DNA vector, which is then inserted into the suitable host organism with the help of the enzyme like restriction endonucleases and ligases. This recombinant DNA vector is then expressed itself into the host organism to give us the desired product. This is the whole process of recombinant DNA technology. In the process of recombinant DNA technology, cloning vector or vector DNA play a vital role, and certain characteristics will make a cloning vector to be a good cloning vector, and these characteristics are presence of ORI site, selectable marker and cloning site. As we all knew that ORI site are those sequences of DNA molecule from where the replication start, and any piece of the DNA when linked with this site will be made replicable in the host organism. Selectable marker are those genes, or you can say sequences of DNA whose expression will help us to eliminate or identify the transformant one from the non-transformant one. And cloning site are those sequences of DNA molecule where our gene of interest is inserted in the cloning vector. Now consider a case where our gene of interest is inserted at such a place where our selectable marker is present in the cloning vector. क्या होगा जब हमारा जीन ऑफ इंटरेस्ट जो है वो ऐसी जगह पर क्लोनिंग वेक्टर के अंदर इंसर्ट होगा जहां पर हमारा सिलेक्टेबल मार्कर प्रेजेंट है. उस केस में हम कैसे जानेंगे कि हमारे सेल्स का ट्रांसफॉर्मेशन हुआ है या नहीं हुआ है? नाउ लेट अस ट्राई टू अंडरस्टैंड दिस विद द हेल्प ऑफ द plasmid vector PBR322. Now let us try to understand the concept of insertional inactivation. Here in this diagram of plasmid vector PBR322, you can see that this is your ORI site from where the replication start. PST1, PVU1, EcoR1, BAM-H1, SAR1 and PVU2 are the various restriction sites or cloning sites that are present in the cloning vector PBR322, and amplicin resistant and tetracycline resistant genes are two selectable markers that are present in this cloning vector. Now consider a case where my gene of interest or foreign DNA is inserted at such a place in the cloning vector where my tetracycline resistance gene or my selectable marker is present. In that case, my selectable marker is not able to express itself, and this phenomena is termed as insertional inactivation. Now let's have a look on the statement of insertional inactivation. The process of introduction of desired gene in the coding region of the DNA that result in the inactivation of an enzyme is termed as insertional inactivation, or in other words we can say that insertional inactivation is the inactivation of gene upon insertion of another gene inside in its place or within its coding sequence. यहाँ पर हमारा जो फॉरेन डीएनए है वो हमारे टेट्रासाइक्लिन रेजिस्टेंस जीन की प्लेस में जाकर इंसर्ट हुआ है, इसलिए हमारा टेट्रासाइक्लिन रेजिस्टेंस जीन जो है वो अपने आप को एक्सप्रेस नहीं करेगा, और इसी फिनोमेना को कहा गया है इंसर्शनल इनएक्टिवेशन. नाउ द क्वेश्चन अराइज, इफ माय selectable marker gene is not able to express itself, then how I will know that my colonies are get transformed or not. अब मुझे ये कैसे पता चलेगा कि मेरी जो बैक्टीरियल कॉलोनीज हैं वो ट्रांसफॉर्म्ड हुई है या नहीं हुई है? Now let us try to understand this. After inserting my recombinant vector into the host organism and culturing it, we get three different type of colonies on the master plate here in this diagram. These three different type of colonies are represented by three different colors. Say the red one will represent the non-transformant or natural bacteria that will show sensitivity towards the antibiotic amplicin and tetracycline, hence easily excluded from the bacterial culture. The second one, the green one, will represent the transformed with non-recombinant vector, as these are those bacterial colonies that will get transformed with the plasmid but do not contain my gene of interest. Hence, they are able to grow on both amplicin and tetracycline containing medium. And the third one, the blue one, will represent the transformed with recombinant vector. These are those bacterial colonies that contain my gene of interest, as my gene of interest is inserted at such a place in the cloning vector where my tetracycline gene is present. So these colonies do not show resistivity towards tetracycline, as my tetracycline resistance gene is not active but inactive due to the insertion of my gene of interest. Now, but they are able to grow on amplicin containing medium. Now, both of these colonies, the green one and the blue one, are able to grow on amplicin containing medium. Then how could I come to know that whether my bacterial colonies get transformed or not? Here we use the replica plating technique to get or to eliminate or identify the transformed from non-transformed one. In replica plating technique, we use a sterile marbling growth and get an imprint of the colonies that are going on an amplicin containing medium and then culturing it on tetracycline containing medium, as colonies that bacterial colonies that contain my gene of interest are not able to grow on tetracycline containing medium. They will not grow on the tetracycline containing medium, but the colonies that get transformed with non-recombinant vector, they will grow on tetracycline containing medium. With this, I am ending this video now. Hope my videos might be useful for you people. So please subscribe to my channel and stay with me, connected with me. Till then, bye bye.